Hi, welcome to Organic Chemistry. Here we're going to talk about epoxides. Epoxides, also called oxiranes and oxides, are functional groups that involve a three-membered ring of carbon, carbon, and oxygen. As you know, one of the main ways of making epoxides is from alkenes and a peroxy acid, such as, for instance, MCPBA. Just recall from previously when we talked about alkenes, this involves the pi bond reaching out to the oxygen and then the oxygen reaching back to the pi bond. So this makes a three-membered ring because the oxygen acts as both electron pair acceptor and electron pair donor. Now mechanistically this actually doesn't happen in a stepwise fashion as I've shown. Instead it happens in a concerted process. All these bonds happen simultaneously, and the oxygen makes connections to either end of the pi bond, making a three-membered ring. You can also make an epoxide by an internal SN2. If I had a halohydrin, that's a species that has an alcohol adjacent to a chloride, or to a halide of any sort, I suppose. And remember, we can make halohydrins from alkenes with, say, Cl2 and water, if you recall. Well, if this thing is treated with a base, it'll make an epoxide. How so? Base can deprotonate the hydrogen from the alcohol to make the alkoxide, which is a strong nucleophile. It would like to do an SN2 reaction on an alkyl halide. Well, right next to it, we've got an alkyl halide, and there we go. This process is no different from an intermolecular SN2 reaction. It's just one end of the molecule reacting with the other, or one side of the molecule reacting with the other side. And so instead of having two things come together to make a connection, you have something reaching around to make a connection to itself, which makes a ring. There are other ways of making epoxides, and I'm not going to spend a lot of time on these. There are metal-catalyzed ways of making epoxides, where the metal and some oxidants, in this case a hydroperoxide, act as an oxygen donor, an oxygen atom donor. And I show some arrow pushing to show how this might happen. But if this catalyst is achiral, the product will be racemic. That epoxide involves chiral centers. But in this case, I would get a racemic mixture. I'd get both stereoisomers. But what if instead we had a chiral agent? Achiral things lead to achiral results. Chiral things lead to chiral results. If I somehow picked out shoes by having them interact with socks, I know it's a dumb analogy, but oh well. Well, socks are achiral, and they interact with right and left shoes equally well. So we'd get a 50-50 mix. On the other hand, if I picked out shoes by seeing how they interacted with my right foot, well, I would get right shoes and not left shoes, because the right shoes interact better with my right foot. If I could have a chiral catalyst, and if I took my titanium and I added in a chiral add-on, in this case diethyl tartrate, I would get a chiral catalyst, which then leads to a chiral result. This is the Sharpless epoxidation, for which he won the Nobel, along with a couple of other important reactions that he developed. Particularly looking at installing chiral centers in molecules. And Sharpless found that he could make either stereoisomer by using enantiomeric additives. This diethyl tartrate, and here's the enantiomer. This one gives this isomer of product. This one gives the enantiomer. Because if I picked out shoes with my right foot, I would get right shoes, but if I picked out shoes with my left foot, I would get left shoes. That is, if my reagent was the mirror image, I should get the mirror image of product. Another way of making epoxides is with manganese. And there are manganese complexes that will make epoxides. They're essentially oxygen atom sources. Well, Jacobson developed a chiral epoxidation catalyst, where he made kind of a chiral piece that sits around the manganese that then leads to a specific stereoselective production of epoxides. Mostly I want you to focus on the MCPDA and the internal SN2. Those others I think are interesting, especially in how they help us start talking about making specific stereoisomers. But let's go on to talk about what do epoxides do. Well, epoxides are very strained rings, and as I mentioned before in the alcohol section, I said we can break those things open, and I said we'll do more on this later. This is actually a slide pulled from that previous presentation, but let's go through those now. If I took an epoxide and I treated it with a strong nucleophile, a reaction would occur to make, well, a product in which I've added in my nucleophile and I've broken open that three-membered ring. Some further analysis might lead me to understand that the OH in the product came from the epoxide oxygen. Well, how can we get there? My ethoxide is a strong nucleophile, and it will add and do an SN2, a backside displacement, breaking open that ring. Usually O- is a horrible leaving group, but in this case this is energetically favorable because we break open that three-membered ring. And then if I lose proton, or if I add a proton, I get my product. But notice my epoxide had two sides, had two carbons, but this specifically added to the less sterically hindered one. But that makes total sense if we think about this mechanism. This is an SN2 process. And SN2 processes don't happen at tertiary positions. They happen best at primary positions. In this particular epoxide, we have a tertiary position and a primary position. And my nucleophile comes to the less sterically hindered one. On the other hand, if we took the exact same epoxide and we treated it with acid and ethanol, we again would break open the epoxide, making an OH and adding this nucleophile. But we get the opposite regioisomer. What's happening here? Again, the OH in the product comes from the oxygen in the epoxide, and the O carbon-carbon came from the ethanol. But here, this is acid catalyzed. If my epoxide reaches out to a proton, we're going to make oxygen with three bonds, a bond. Carbocation intermediate would get attacked by some weak nucleophile, ethanol in this case. Loss of a proton leads to product. But notice here, the regioselectivity of epoxide ring opening is such that it gives the more stable of the two potential carbocations. So this gives kind of the opposite selectivity of the previous one, but it's because this is an SN1 type mechanism. We break a bond, we form a carbocation intermediate, and then our nucleophile comes along. That's an SN1 process, as opposed to the SN2 process in the other one. And as you recall, SN1 and SN2 have sort of opposite requirements as far as substrate. SN2 work best on primary positions, and they don't work at all on tertiary. SN1 work best on tertiary, and they don't work at all on primary. We see the same trend here. There's some other reactions of epoxides. For instance, they can react with triphenylphosphine to make alkenes. Triphenylphosphine can act as a nucleophile and break open that epoxide, but the O- can latch on to the phosphorus to make a phosphooxetane. And this should look familiar, because when we talked about the Wittig reaction, we formed a phosphooxetane intermediate, and then that broke down to make an alkene. And the same thing is true here. We can actually use this in a two-step process to change an alkene stereochemistry. If I took an alkene treated with MCPDA, I'd make an epoxide. And this is a syn addition. We would retain the stereochemistry. The two methyl groups are trans to each other. But if I treat this with triphenylphosphine, I end up with the cis alkene because of this rotate step. When I rotate around this bond to get the oxygen near the phosphorus, this carbon group ends up down. All right. Epoxides have an interesting chemistry when it comes to biological systems. For instance, this compound is squalene oxide. Squalene is a terpene. That is, it's a compound that the carbons are all derived from these isoprene, these little 5-carbon pieces. And squalene can be broken down, here I've used color coding, to see a bunch of little of these pieces. Well, they've got a 4-carbon chain with 1 methyl branch. 4-carbon chain with 1 methyl branch. And here, and here, and here, and here. Notice I switched the orientation. These are head to tail, and then tail to tail, and then head to tail, head to tail, head to tail. Squalene oxide is important because under some certain enzyme catalyzed conditions where there's acid around, they are transformed into I'm totally blanking on the name of this compound, but it doesn't matter. It's a step on the way to making cholesterol, which is important. You make cholesterol biosynthetically. You don't just get it from eating too much fast food. You need cholesterol because it's a key component of your cellular membranes, and it also is a precursor to important hormones in the body and other compounds. Anything with this 6-6-6-5 ring system, it's called a steroid ring system, ultimately comes from this cyclization. You can have too much cholesterol. That's what happens when you eat too much fast food, and it can build up in your veins, arteries. But that's not really our point right now. What I want to focus on is how in the world does this happen? It kind of looks like magic. But if I were to draw a different conformation of the squalene oxide, well, I can see some relationship there. If this is catalyzed by acid, the epoxide can get protonated. Now I get oxygen with 3 bonds. Let's break one to make a carbocation. But that carbocation needs electrons, and right next door I've got this alkene, which has electrons. So let's imagine that reaching over. But that's going to make a new carbocation. Okay, let's show it again here. But if I've got another alkene, that can reach over to form a carbocation, and an alkene can reach over to form a carbocation, and again. We're not quite done. We're not quite at Lannisterol. That was the name of that compound that I was forgetting before. To get there, we have to do a couple of hydride shifts, a couple of methyl shifts, and then a loss of a proton to form a pi bond. And this is all stuff that's normal for carbocation chemistry. But it started out with an epoxide. One other really brief epoxide story. Epoxides are reactive towards strong nucleophiles. If you smoke, which you shouldn't, you're going to be exposed to benzoate-pyrene. You can also find this in other environmental contaminants. Benzoate-pyrene, your body wants to get rid of it because it's not a nutrient, and so it will start to oxidize it. There's a series of enzyme-catalyzed oxidations in the process making this epoxide. The reason your body starts to oxidize organic compounds is to make them into something more soluble so they can be excreted via the urinary tract. But this epoxide that's formed is susceptible to attack by nucleophiles, like your DNA. Your nucleic acids have all these lone pairs that can act nucleophilically, like this N7, like this nitrogen. It's not 7, it's a different one. Sorry. Can act as a nucleophile and break open the epoxide. Why is that an issue for us? Well, we have now made a covalent attachment between this huge molecule and our DNA. So here we see a structure of benzoate-pyrene covalently bound to a nucleic acid base. But this totally messes up the structure of your DNA. And it's going to mess up transcription, translation, and replication. I won't go through this slide, but it causes, for instance, frame shifts, which means it's like when you take one base out of a DNA sequence, and everything shifts over 1/3 of the codon, and that really messes things up and will lead to long-term problems for a living cell. Alright, that's really all I have to say about epoxides. We really make them two ways that are important to us. The MCPBA with an alkene, and then the internal SN2 when you've got a halohydrin that you treat with base. Epoxides want to react by ring opening, and so anything that's a nucleophile will probably help pop open that epoxide. Acid catalyzed reactions will help facilitate that epoxide opening as well. Alright, that's it. Thanks for listening, and I will see you soon.